Good Monday morning. I hope you guys are doing great today. It's a little chilly outside. Hopefully you're bundling up and staying warm. We got a crazy week this week. So let's get started. First of all, on the front stream, I have put reminder that there's no school tomorrow for election day. Encourage your parents to get out and vote. And then Wednesday, we have a half day. So our schedule is right here that I've attached for you for your half day. All right, for our classwork, I have our lesson plans up here. So let's click into our lesson plans. All right, so today, Monday, you're supposed to watch the Amoeba Sisters and do the DNA versus RNA worksheet. Just do the front of that worksheet, the first part of that worksheet. We're also reading through Chapter 12, Notes, Section 2. And then we're going to finish our DNA model. So that's kind of what we're going to work on this week. You can see Tuesday we have no school. Wednesday is a half day. We're going to watch another Amoeba Sister video and do the worksheet that goes with it on DNA replication. And then I've got a quiz for you in the Google Classrooms when you kind of come back in here. So this is the video we're going to do today, and the worksheet's in there too. Chapter 12 notes we're going to work on today. Your study guide for section 2. Now you also received this last week, so this one is just section 2 is what we're going to work on. The DNA, RNA, this is for Wednesday. And then Thursday, we have a DNA replication, and then we're doing our quiz. So we have all these things to do. Please be reading chapter 12 in your textbooks. You all have a textbook at home. So make sure you're clicking through flipping through and reading your actual textbook. Reading for information is very important. And that's chapter 12. All right, we're going to click into some notes real quick. So our Amoeba Sisters video today is more about DNA, RNA, and about what you do with that DNA. So it's a little bit different. It doesn't quite go over what we're teaching just today and then the rest of this week, but it gives you a good idea to go back over the parts of DNA and get a little preview about RNA. The rest of this week, we're going to be talking about DNA replication. Let me grab my model. Okay. Hopefully you have your model at home. If you have made it on paper, you've cut and colored, somehow you have your model at home. And even if you just wanted to write on a piece of paper, sugar, phosphate, and then do a few of these nitrogen bases in the middle, just so you have an idea. You'll notice that one side is right side and the other side is upside down. You're like, what's up with that? Why is this upside down? Well, that's because DNA is not identical on both sides. It's complementary. Complementary means that they complement each other. So the G pairs with the C, and the A pairs with the T. They're not identical, but it's their partner on the other side that makes up the DNA. And so we call this complementary pairs, okay? And whenever we read the DNA code, so I'm going to give you these 3 codes right here. So this is C, T, and G. When our body wants to read DNA, it's going to come in with an enzyme, and it's going to read C, T, G. It doesn't read the other side. It just reads the 3 code on what we call the leading strand. These 3 letters correspond to an amino acid that builds together a protein. So we're just reading off of one side. We also are going to need to make a copy of our DNA. So when we make a copy, you can't copy both sides the exact same because they're not the exact same. So we need to make a copy of our DNA. And so let's talk about why we need to make a copy of our DNA. What's the point of this? Well, there's 3 main reasons. One is for growth. My fingernail right here is growing, and I need to make more fingernail cells. My cuticle down here needs to make more fingernail cells. So my fingernail's always growing. My hair is always growing. Some of you are growing taller and bigger as you're growing. I'm old, so I'm not growing any taller, but I can definitely grow wider. So for growth, we need to make more cells. And in each cell has to be a copy of that DNA. It needs to code for my proteins and my information in every cell of my body. So growth. The second thing is repair. I got attacked by my cat this weekend. My cat scratched the fire out of my foot, tore open my foot, and I was bleeding, and it was gross, and the cat got kicked. It's okay. We're all okay. But I need to regrow some new cells on my foot and some new blood cells. So I need to repair, which is number 2. I need to repair my cells so that my boo-boo is not there anymore. So for repair, have you ever gotten injured before? Well, your body had to repair and make a new cell to fill in that damaged area. All right, and then the third one is reproduction. Reproduction. So when we are going to make an offspring, we need to make a copy of our cells to make that offspring. One day you are going to grow up, graduate college, find someone that you love, get married, and decide to have children in that order, and you'll reproduce. Boys, half of your DNA is in a sperm cell, and it's got only half of your genetic information. And girls, half of your genetic information is in an egg, which is in the ovary, which is part of the female reproductive system. And the egg and sperm will come together and make one cell with that one set of DNA for your genetic code. But that DNA has to be created. So for reproduction, you'll have to put half of this DNA inside of one of your reproductive cells. Luckily, you don't have to think about this, and it'll happen automatically. Okay, so in order to make new DNA, it's complicated, and your body's doing this all the time. This is a good reason why we should take good care of our body. We should rest and get sleep and help our body to not be stressed out and exhausted so that it can make new DNA and make it as good as it's supposed to do. All right, we're going to call it semi-conservative replication. So let's break this word down in our Latin. Semi means a part. Okay, semi is like a portion of it. So semi is a portion of my DNA. To conserve means to hold back. So when I'm going to make a new copy, I'm going to conserve part of the DNA in my replication. That sounds kind of odd. All right, the parental strand of DNA separates and serves as a template to produce new DNA molecules that have one strand of the parental DNA and one strand of the new DNA. So let's look at this. The parental strand is here, and we're calling the parental strand is just the original DNA. So where my cat scratched me, I have where my original DNA is on my skin cells. My skin cells need to make a new copy of the skin cells. They need to make a new copy of the DNA first. So the first DNA that's in that cell of my body right now, that DNA strand is what we're going to call the original parent strand. That parent strand is going to open up and then make new copies. But look here, half of it is old, the parent strand, and half of it is new. And same thing here, my new strand of DNA is really half old and half new. Let me go back to my model here. If I cut this up, but then made a new pair based off of this, so my old is binding to a new A, and my old A is binding to a new T. So I'm making two strands off of this. Thinking, why would I use half of the old? Well, why do you think we would use half of the old strand to match up a new strand? Well, here's the reason. Have you ever copied from somebody and you've gone back and forth writing and you've looked and you wrote and you looked and you wrote? Have you ever messed up? Well, our body can't mess up. If we get a mistake, that means the amino acid gets changed. That means the protein gets changed. That means I get changed. Little changes could give me like a weird coloration on part of my skin. Maybe I have a weird hair color coming in. Most of the time they're unaffected. but sometimes that could happen where something mutates and goes wrong. And I could end up having a cancerous cell or maybe a tumor or just kind of a lump. Anyways, I don't want to mess up. So instead of copying back and forth, our body uses this as half of the original. So I'm less likely to make a mistake. I make the new copy using the original one. So I'm less likely to have a oops, because oopses are bad. All right, so first the DNA has to unwind. So when you unwind, you're going to use a helicase. Now listen, helicase is an ACE, that's an enzyme. So remember, what are some facts about enzymes? Do you remember that enzymes are reusable, that they're catalysts, they speed up chemical reactions? You remember that enzymes are a protein? You remember that enzymes bind to specific substrates at the active site? Do you remember they're affected by temperature, pH, enzyme concentration? Good. All right, helicase comes in and he's going to unzip or unwind the DNA. RNA primase, here's our second guy, primase, is going to add a short segment of RNA. This is why we watched the video, The Amoeba Sisters, first, to kind of talk about RNA. So RNA is going to code. So let me go back to my model here. Okay. So let's just fold this in half because we're just going to be looking at half of it right now. Okay. All right. This is a T. T binds with A. This is a G. G binds with C. Okay. But this is the one I want to look at down here. T for DNA. For RNA, it would bind with the U. So the RNA primase, A binds with U. The thymine gets replaced with uracil on RNA. There's no thymine on RNA. It's only uracil. So A would bind with U. So the RNA primase puts down a little code of RNA to prime up, saying, here's my starting position. Everybody get to work. All right, DNA polymerase. This is our smart guy. He is the one who does most of the work. DNA polymerase is going to come through, and he's going to be working towards the 3' end of our DNA. All right, so here he is working. So let's take a look at all of this again. Here's my parent strand of DNA. Here's the helicase that's unzipping and unwinding, breaking those hydrogen bonds and unzipping. All right, here is my DNA polymerase. Remember, the RNA primer or primase has laid down a little RNA code, and then the DNA polymerase can get to work. Now, on one side that's going in the 3' direction, he can be able to continue this very quickly. It's just boom, 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 boom. As the helicase unzips, he just keeps laying down more code. Remember, semi-conservative, meaning half old, half new. All right, on the other side, though, we're in a pickle. All right, the other side doesn't run in the 3 direction. It runs in the 5 direction. So what is RNA polymerase to do? RNA polymerase only goes to the 3' end. So on the other side, he's going to start here and go up, and then he's going to move down and go up, and then he's going to move down and go up. So let's look at it here in the picture. The DNA polymerase is going to do this section, and then he's going to come up, and he's going to do another section and come up, and then do another section and come up. So he's still moving towards the 3, but you can see it's in all these little fragments, okay? One strand is called the leading strand and is elongated as the DNA unwinds. The other strand is called the lagging strand, and it elongates away from the replication fork. The lagging strand is called the Okazaki fragments. Let's all say that. Okazaki. Okazaki. What do you think Okazaki fragment is named from? Dr. Okazaki. I'm telling you, if I ever discover something, instead of Chargaff's rule, it'll be Hunt's rule. Instead of Okazaki fragments, it could have been Hunt fragments. Some of these people are a little conceited, putting their names on everything. But I think I might be too if I discovered something awesome. Okay? All right, finally, the DNA polymerase removes that RNA primer, and he fills in the places with the DNA nucleotides. The DNA ligase is our last of our enzymes. There's only 4 that's going to link the 2 sections together, okay, in the Okazaki fragments. All right, so let's go back through here. On the lagging side right here, this is the ligase who glues those little Okazaki fragments. Remember that the DNA polymerase works in Okazaki fragments as he comes through here, laying down the complementary base code. Okay? All right. When you look at how eukaryotes replicate versus prokaryotes, it's night and day. Eukaryotes are going to unwind the DNA in multiple areas, so that huge strand of DNA is coming through at different points all at the same time. It's like a mob attacking DNA and undoing it all at the same time. For prokaryotes, no. Prokaryotes are circular DNA. It starts in one point and goes around the circle, and it's done. All right, remember, prokaryotes only have one circular DNA. We have 23 chromosomes in our body with 23 long, complicated things of DNA. It takes a little bit of time to copy all of our DNA. It doesn't take long for bacteria, which is why bacteria reproduce really quickly. That's why you went to bed feeling fine and woke up with strep throat, because that bacteria reproduced all night long and now you're sick. Okay? All right. 1, most of this won't make much sense just listening it to me one time. 2, you need to read the book, chapter 12, section 2. 3, when you watch the next Amoeba Sisters video, they do a great job in breaking down these enzymes and giving you some good videos. You will understand it more. And 4, there's a whole new DNA replication sheet we're doing Thursday also that goes back over all this again. I think it'll help you out. You have to read that book and study. All right, let me exit this. Okay, so the rest of the period or at home, you're going to work on the Section 2 study guide. This will be for Wednesday. And then Thursday, we've got this DNA worksheet and then the DNA quiz. Okay, again, on Fridays, I have Google Meets. I've had Google Meets every Friday. Usually just depends on my schedule, but it's around 10:30. We'll have a Google Meet. I try to get you plenty of time ahead of time. If you need to meet with me or talk to me on Fridays, that's a great time to talk. Ask me questions. But remember, I'm at school until 3:30 and then I'm at home. So use time of school time to get school work done. Please continue making up any missing assignments. Last week, we kind of got caught off guard with the bad weather on Thursday. I would really love to get all makeup work done and all your grades caught up and back to good grades like they should be. All your work should be turned in on Fridays. If you have to use a weekend to get your work done, you can, but then it'll be due on Monday. So make sure you're turning in your work. I'm trying to put in grades on Infinite Campus that show you what I'm going to take a grade on so that you know this is an assignment you have to do, but you have to get your work done each week and keep up with the class. I hope you have a great week. Hope you enjoy your election day off of school and stay warm because it's getting cold outside. Have a great week.